Hey there, I am Maruk Said, a microbiologist and a science educator. Science is my passion and I'm here to share it with you. Microbiology and molecular genetics, all relevant definitions. Let's learn about molecular genetics, all relevant definitions. Molecular genetics. Molecular genetics is a branch of genetics that focuses on the study of the structure and functions of the genes at a molecular level. It involves analyzing DNA, RNA, and proteins to understand how genetic information is stored, replicated, and expressed. DNA, deoxyribonucleic acid. DNA is a molecule that carries the genetic instructions used in the growth, development, functioning, and reproduction of all non-living organisms and many viruses. It consists of two strands of nucleotides, twisted into a double helix. Gene. A gene is a segment of DNA that contains the instructions for producing a specific functional product, usually a protein or RNA molecule. Genes are the basic unit of heredity and determines the trait and characteristics of the organism. RNA, ribonucleic acid. RNA is a molecule that plays a central role in various cellular processes including gene expression and protein synthesis. There are several types of RNA including messenger RNA, transfer RNA, and ribosomal RNA. Genetic mutation. A genetic mutation is a permanent change in the DNA sequence of a gene. Mutations can be beneficial, neutral, or harmful, and they are the source of genetic diversity in population and evolution. Genetic engineering. It is the manipulation of organisms' gene using a biotechnology techniques. It involves the insertion, deletion, or modification of DNA to alter the characteristics of organism or to produce specific products. Polymerase chain reaction (PCR). It is a laboratory technique used to amplify a specific segment of DNA, making it easier to study or analyze. It is widely used in various applications such as DNA sequencing, gene cloning, and diagnostic testing. Genetic code. The genetic code is a set of rows by which the information encoded in DNA or RNA is translated into the amino acid sequence of proteins during protein synthesis. Gene expression. Gene expression refers to the process by which information from a gene is used to synthesize a functional gene product such as a protein or RNA molecule. Genome. A genome is a complete set of an organism's genetic material including all of its genes and non-coding sequences. Genetic variations. Genetic variation refers to the differences in the DNA sequences among individuals of the same species. It is a fundamental aspect of evolution and plays a role in determining an individual's traits and susceptibility to diseases. Recombinant DNA. Recombinant DNA is a DNA that has been artificially created by combining genetic material from different sources, often from different organisms. It is a key technique used in genetic engineering. Genotyping. Genotyping is a process of determining the genetic makeup of an individual or organism by analyzing their DNA, typically through methods like PCR or DNA sequencing. Gene editing. Kindly support my channel by subscribing to this channel. Gene editing is a set of techniques that allow scientists to modify an organism's DNA, enabling precise changes to be made to the genome. CRISPR-Cas9 is one of the most well-known gene editing tools. Transcription. Transcription is a process by which an RNA molecule is synthesized from a DNA template, producing messenger RNA that carries the genetic information from the nucleus to the cytoplasm for protein synthesis. Genetic locus. A genetic locus is a specific physical location of a gene or DNA sequence on a chromosome. Allele. An allele is a variant form of a gene that arises due to genetic mutations and occurs at a specific locus on a chromosome. Different alleles can lead to variation in traits or characteristics. Genotype. The genotype refers to the genetic makeup of an individual or organism, including the alleles present at specific loci. It determines the heredity potential of an organism. Phenotype. The phenotype is the observable physical and functional characteristic of an organism resulting from the interaction of its genotype with the environment. Gene regulation. Gene regulation refers to the mechanism that controls the expression of genes, determining when and to what extent a gene is transcribed into RNA and translated into a protein. Genetic recombination. It is a process by which genetic material from two different sources, usually homologous DNA molecule, is exchanged, leading to the production of a hybrid DNA molecule. Transcription factor. A transcription factor is a protein that binds to the specific DNA sequences near genes and regulates the transcription of those genes, influencing their expression. Gene silencing. It is a suppression or downregulation of gene expression, preventing a particular gene from being transcribed into RNA or translated into a protein. Genetic mapping. It is a process of determining the relative positions of the genes or genetic markers on a chromosome. It helps study the inheritance pattern of the specific traits and the genetic basis of diseases. Genetic screening. It involves testing individuals for specific genetic conditions or predisposition to identify potential risk or provide personalized medical care. Gene therapy. It is a therapeutic approach that involves introducing or modifying genetic material into a person's cells to treat or prevent genetic disorders or diseases. Genetic counseling. Genetic counseling is a process that provides information and support to individuals and families regarding the risk of inherited genetic conditions and the options available for managing or preventing them. Reverse transcription. It is a process of synthesizing DNA from an RNA template. It is a crucial step in the replication of retroviruses like HIV and in the creation of complementary DNA, cDNA from RNA for various molecular biology techniques. If you have reached till here, don't forget to support this channel by subscribing. Genetic linkage. It refers to the tendency of certain genes located close together on the same chromosome to be inherited together during reproduction. Genetic linkage is the basis of genetic mapping. Epigenetics. It is the study of changes in gene expression that occur without altering the DNA sequence. Epigenetic modifications can be heritable and can influence an individual's trait and susceptibility to disease. Gene expression profiling. It involves analyzing the activity of multiple genes simultaneously to understand the patterns of gene expression under specific conditions or in different cell types. Transposons. A transposon, also known as transposable element, is a mobile DNA sequence that can change its position within the genome. Transposons can have important evolutionary and genetic consequences. Restriction enzyme. A restriction enzyme is an enzyme that can recognize specific DNA sequences and cuts the DNA at or near these recognition sites. Restriction enzymes are commonly used in molecular biology techniques such as DNA cloning and genetic engineering. Polymerase. A polymerase is an enzyme responsible for synthesizing new DNA or RNA strains using a DNA or RNA template. Different polymerases are involved in DNA replication, transcription, and various molecular biology procedures. Genetic drift. It is a random change in the frequency of gene variants alleles in the population over successive generations due to chance events rather than natural selection. Genome sequencing. Genome sequencing is a process of determining the complete DNA sequence of an organism's genome. It has become increasingly accessible and plays a vital role in various fields including personalized medicines and evolutionary studies. CRISPR-Cas system. The CRISPR-Cas system is a powerful gene editing tool derived from a bacterial defense mechanism against viral infections. It has revolutionized genetic research and holds immense potential for therapeutic applications. Genetic transformation. Genetic transformation is a process by which foreign DNA is introduced into a cell, resulting in a stable and heritable change in the cell's genetic makeup. Gene amplification. It is the increase in the number of copies of a specific gene or DNA sequence within a cell. Amplification is a common phenomenon in cancer cells and certain genetic disorders. Gene expression regulation. It involves the control of gene activity either by activating or repressing transcription. It plays a crucial role in developmental processes and cellular responses to environmental cues. Genetic disorder. A genetic disorder is a condition caused by abnormalities or mutations in an individual's genetic material, often leading to specific health problems or developmental issues. DNA sequencing. DNA sequencing is a process of determining the precise order of nucleotides A, T, C, G in a DNA molecule. It is essential for studying genes, genomes, and genetic variation. Genomic medicine. It is an approach to medical practice that uses information about an individual's genes and genetic variations to guide personalized healthcare and disease management. Pharmacogenetics. It is a study of how an individual's genetic makeup influences their response to specific drugs. It aims to optimize drug therapy based on the person's genetic profile. Gene drive. It is a genetic phenomena that can increase the likelihood of an specific gene or genetic modification being inherited and spread through population. It has implications for controlling disease vectors and invasive species. Genetic marker. A genetic marker is a known DNA sequence with a known location on a chromosome. Genetic markers are used in genetic mapping and linkage analysis to track the inheritance of specific genes or traits. Knockout mouse. A knockout mouse is a laboratory mouse in which a specific gene has been intentionally inactivated or knocked out. These mice are used in research to study the function of genes and their role in disease. Epistasis. It is a genetic phenomenon where the effect of one gene is dependent on the presence of one or more other genes. It can influence the expression of traits and their inheritance pattern. RNA interference. RNA interference is a biological process where small RNA molecules regulate the expression of specific genes by targeting complementary messenger RNA sequences for degradation or inhibition. Genomic imprinting. It is an epigenetic phenomenon where specific genes are expressed in a parent of origin specific manner due to epigenetic modification. Recombinant protein. It is a protein produced through genetic engineering techniques by inserting the gene encoding the protein into a different organism or expression system. DNA methylation. It is epigenetic modification where methyl groups are added to cytosine bases in DNA, influencing gene expression and regulation. RNA splicing. RNA splicing is a process in eukaryotic gene expression where introns, non-coding regions are removed and exon-coding regions are joined together to form mature messenger RNA before translation. DNA repair. DNA repair mechanisms are cellular processes that correct damage to the DNA molecule, maintaining the integrity of the genetic material. Nucleotide excision repair. It is a DNA repair mechanism that corrects various types of DNA damage, including bulky lesions caused by UV radiation. DNA profiling, DNA fingerprinting. It is a technique used to analyze an individual's DNA to identify unique genetic markers for forensic, paternity, and identification purposes. Restriction, fragment, length, polymorphism. It is a molecular biology technique that involves cutting DNA molecules with restriction enzymes, resulting in DNA fragments of varying lengths, which can be used for genetic analysis and mapping. Genome-wide association study. It is a study that examines the entire genome of a large population to identify genetic variations associated with particular traits, diseases, or phenotypes. Codon. A codon is a specific 3-nucleotide sequence in messenger RNA that encodes for a particular amino acid during protein synthesis. Epigenome. It refers to all the chemical modification to the DNA and associated proteins that can influence gene expression without altering DNA sequence itself. So that's it for today. Thank you so much for watching till the end. Don't forget to subscribe to my channel.